Are there any other questions about what we discussed yesterday or earlier this week before we move on to some new material? You can't think of anything right now and you think of something later, feel free to interrupt me. You know, I know some teachers don't like to be interrupted, but I think it's something that can be useful for us if you need clarification about something. So let's see. Okay. So you should be able to see a document called Hardy-Weinberg equation. Let me know if you're not seeing that and I've got the wrong document up or something else is going wrong. So remember what we said the other day. Mendel didn't publish his work on genetics until 6 years after Darwin published his theory of evolution. And Mendel's work was really lost to science for 35 years until 1900. Nobody realized its significance. So because of this, evolutionary theory was never really updated. So evolutionary theory until after 1900 didn't include the most relevant ideas about inheritance and genetics. Mendel's work was never incorporated into Darwin's work. And in the first half of the 20th century, evolutionary biologists who were, in a sense, applying Darwin's ideas, and population geneticists who were applying Mendel's genetics principles to study populations started to work together. And what they produced was eventually called the modern synthesis. And it combined Darwin's theory of evolution and Mendelian genetics. And really, the Mendelian genetics is basically the same as we talked about in Biology 105. But instead of applying it to an individual mating, we're going to focus on thinking more in terms of an entire population. And if you remember, even back in Biology 105, we said that when you think about a Punnett square isn't a prediction for one particular mating. It's a prediction for what we see when we have a lot of matings like this. So we haven't really talked about population genetics before. We talked about molecular genetics. We talked about Mendelian genetics, things that follow Mendel's principles. We talked about non-Mendelian genetics, things like X-linked characteristics that don't follow Mendel's rules. Population genetics studies genetic differences. Now, it studies two types of genetic differences. Genetic differences within a population. So that would mean if we're talking about people, for instance, we could study the differences, the genetic differences between people in the United States. But it also studies genetic differences among populations. And so this is genetic differences in different populations. So this would be studying genetic differences between, say, people in the United States and people in Canada. Keep in mind when we look at natural populations, not always, but usually they're in a much smaller geographic area than the United States or Canada. And what population genetics does is it extends what we learned about Mendelian genetics to complete populations. And basically, when we look at population genetics, it's mathematical in nature. So one of the things that you might have noticed on the syllabus was that it said you would need a calculator. So if your phone doesn't have a calculator as one of the apps that comes with it, you might want to download a calculator, or if you're doing this on your laptop or computer, and same thing, it doesn't have a calculator app, you might want to download one, because starting next week, we will need a calculator for some of the things that we're doing. Now, keep in mind, your calculator doesn't have to do anything real fancy. He has to do the 4 basic operations, obviously: addition, subtraction, division, and multiplication. Beyond that, you should be able to square numbers and take square roots with it, but that's really the only functions that you're going to need. So pretty much all calculators do the 4 basic operations, but they don't all do squaring numbers and taking square roots. But that's actually quite a bit of what we'll be doing. So you definitely want one that does that. You don't want to have to do that longhand. And so let's start thinking about this Hardy-Weinberg equation. And to think about Hardy-Weinberg and how we can use it to study the genetics of a population, we have to think about something called the gene pool. And the gene pool is technically all the alleles in a population. So notice when we say all the alleles, if we're talking about people, that means the alleles for color blindness, the alleles for Huntington's disease, the alleles for albinism or normal skin pigmentation, the alleles for what color your eyes are, the alleles for what type of blood you have. Now, although that's the gene pool, technically, when we talk about the gene pool, we're almost always going to focus on one allele and one gene at a time. So maybe we want to look at the gene for a human blood type. So remember, there are 3 different genes, or 3 different alleles, I should say, for blood type. Remember, there's an allele that causes type O blood, an allele that causes type A blood, and an allele that causes type B blood. So when we look at population genetics, we're generally going to focus, and probably, let's change the wording here. Let's say we're going to focus on one gene at a time. That's probably a better way to think of it. So here we have a diagram of a gene pool, and we have a bunch of ovals. Each oval represents an individual. And notice that the individuals have X with subscripts in them. So this is not an excellent characteristic. They're kind of using X like we do in math as a variable to indicate, something else. And there's 3 different types of X's in here. They're different colors conveniently. So there's some alleles that are X1, there's some alleles that are X2, and there's some alleles that are X3. So each of these ovals is an individual. So the individuals that are solid colored are individuals that are homozygous. Individuals that are half one color and half another color are individuals that are heterozygous. So this is one way we can think about the gene pool. Now notice we're only looking at whatever gene X is the instructions for. So we're not looking at all the alleles for every characteristic. And the reason is when we start trying to do the math involving for anything really more than 2 characteristics at the same time, the math is just overwhelming. Even computers literally have to spend years trying to solve equations as soon as we have 3 different genes involved. So we're going to stick to one allele. If you have access to a high-powered computer, you could work some problems with 2 alleles, but it gets much more complicated. Now, in a sense, this represents our population. Notice that it can be very difficult to get this information because if you think about something that we talked about back in Biology 105, when we're looking at individuals that show a dominant trait in their phenotype, we don't really have a good feel for whether those individuals are homozygous or heterozygous. If you remember, one of the things we said in Biology 105 was, if someone shows a dominant trait in their phenotype, there's really only one basic way that, short of genetic testing, that we can use to figure out if you're homozygous or heterozygous. So remember, we talked about, for instance, brown pigmentation, brown eyes, and we said that was caused by a dominant genetic allele. Blue eyes were caused by a recessive allele. So if you have brown eyes, how do we know if you're homozygous for brown eyes or heterozygous? Well, unless we want to spend a lot of money and sequence your DNA, we have to look at your family tree. If you have brown eyes, but one of your parents, your mom or your dad, it doesn't really matter which one, but if you have brown eyes and one of your parents had blue eyes, we know that you're heterozygous because you inherited an allele from that parent for blue eyes. Or the reverse is true in terms of generational thinking. If you have brown eyes and we look at your children and one of your children has blue eyes, we also know you're heterozygous because blue eyes are recessive. So someone with blue eyes has two recessive alleles. They inherited one from their mother, one from their father. So if this is your child, you must be heterozygous. Now, the problem is you could be heterozygous even if both of your parents have brown eyes. Because remember, a brown-eyed person could be heterozygous. So if your mother has brown eyes, she could have one allele for brown eyes, one allele for blue eyes. You could inherit her allele for blue eyes. But if you inherit an allele for brown eyes from your father, you'll still have brown eyes. So for people who show the dominant characteristic, it can be very difficult to figure out exactly what their genotype is. We want to keep this in mind because later when we work problems, something I'll say now, and you'll probably hear me say a couple more times, you'll probably hear me say it so many times, you'll get sick of it. We're always going to start with people who show the recessive characteristic in their phenotype. Or if we're studying chipmunks or we're studying corn plants, whatever we're studying, we're going to start with organisms that show whatever the recessive phenotype is. And the reason is because if you have a recessive phenotype, we know your genotype. You have to be homozygous recessive. That's the only possibility, because as soon as you have one dominant allele, you would show the dominant characteristic. You wouldn't show the recessive trait anymore. So we want to keep that in mind. When we work our problems, once we start doing some math, we're always going to start with individuals that have the recessive phenotype. And the reason we have to think about phenotype is because, remember, if I look at you, I can see whether you have blue eyes or brown eyes. If I look at you, I can't see your genotype. So we always have to start with the phenotype, and then we're going to use the phenotype to figure out the genotype. And we're always going to start with the recessive trait showing in the phenotype. Now, when we think about the gene pool, usually we don't show each individual for some reasons that we'll discuss later. Usually what we want to think about is the individual alleles. So we want to think about all the X1s and all the X2s in this population and all the X3s. We don't really care about the complete genotype that they're in. And we'll go back to the text material above this in a moment, but so let's think about the gene pool for fur color in mice. So in this population, we have two alleles. There's an allele that causes black fur color. There's an allele that causes brown fur color, and they've conveniently colored them, well, kind of grayish and brown. So if we look at all the individuals in our population, now notice in this case, they're giving us some information to start with. 48% of the mice in this population are heterozygous black. Well, notice if they're heterozygous black, which characteristic is dominant? Can anybody tell me which characteristic is dominant if heterozygous mice have black fur? Notice we have 16% that are homozygous black. We have 36% that are homozygous brown. But if heterozygotes are black, does that mean black is dominant or does that mean brown is dominant?
Black.
Black. If something shows up in a heterozygous organism, that something is the dominant allele, because remember, That's our basic definition of dominant. A dominant allele is one that always shows up in the phenotype, assuming you have it, of course. If you don't have it, it doesn't show up. The recessive allele can be hidden in the phenotype. Remember, that's what Mendel found somewhat confusing when he did his early experiments. He had those true breeding lines, and one characteristic disappeared when he crossed them with another true breeding line. So in this case, black is the dominant characteristic. Notice if you were just looking at these mice, if you hadn't done genetic analysis, or if you hadn't looked at the family trees of these mice, you wouldn't know that black was dominant. So what you would actually know if you just looked at them, you would say 36% of these mice were brown, and you would say 48 plus 16%, 64% are black. Now, since brown is the recessive characteristic, remember what we said. We would start thinking about this problem in terms of brown. But before we do that, let's think about how we could calculate the allele frequencies given this sort of information. Like I say, a lot of times we wouldn't have something this detailed. We would just know that 64% of the mice had black fur. But let's think about how we would calculate this. And when we talk about how we're going to describe the alleles, we're going to describe what's called the allele frequency. And when we talk about the frequency of an allele, mathematically, the frequency is an allele between 0 and 1. So it can't get smaller than 0, it can't get bigger than 1. And the reason is because 0 and 1 really, in a sense, you could think of these as being percents. 0 is 0% of the population. Well, I know in math we can have negative numbers, but if we're talking about our mice, we could look at a population and 0% of the mice have black fur, but we can't have minus 2% of the mice have black fur. So 0 is the smallest number that we can have. 1 is the largest number we can have because 1 represents 100%. And again, in mathematics, I know you can potentially get a number bigger than 100%, depending on the type of problem you're working. But realistically, if 100% of the mice in our population have black fur, that's everybody. We can't have 101%. That doesn't make sense when we're referring to a real population. So notice 0.5, if F was 0.5, that would mean half of the alleles are the allele for black fur or brown fur, whatever we happen to be talking about. So let's use the numbers that this problem gave us, and then we'll talk about how we would solve this if we didn't know these numbers. So let's think about a population of 100 mice. Since they gave us percentages, we know that mathematically, when we're thinking about mice, a percentage is just how many out of 100. So 48% of our mice were heterozygous. So that means that if we look at these mice, every mouse who's heterozygous has one allele for black fur. So 48% were heterozygous, so that means there's 48 alleles for black fur. 16% of the mice in this population were homozygous for black fur. So if we look at these 16%, if they're homozygous for black fur, they each have 2 alleles. So 16 times 2, that's 32. So we have 48 alleles for black fur in the heterozygous mice. We have 32 alleles for black fur in the homozygous black mice. Notice there are no alleles for black fur in the homozygous brown mice. So we can add 48 plus 32. This means there are 80 alleles for black fur in our population. Now, we can do the same thing for brown fur. We have 48 alleles for brown fur in the heterozygotes. Notice this is the same as black fur because Heterozygotes, by definition, have one allele for black fur, one allele for brown fur. And we have 72 alleles for brown fur in homozygous brown mice, because it tells us there are 36% of these. So 36 times 2 is how many alleles we have in the homozygous brown mice. So we have 48 plus 2, we have 128 alleles for brown fur. So now we're going to add our 80 and 120. That gives us 200 alleles. Now notice, if you were working a problem in this fashion, you can check your work at this point. Because if we have 100 mice in our population, cells in a somatic part of the body of a mouse, so anything other than the eggs or the sperm, somatic cells are diploid. They have the 2n number of chromosomes. So if we have 100 mice, we have to have 200 alleles for fur color, because each mouse has 2 alleles. Each mouse is a diploid organism. So what's the frequency of the allele for black fur? Well, we can take 80, the total alleles, and we can divide by 200, the total alleles. And if we do that, we get 0.4. What's the frequency of the alleles for brown fur? Well, we can take the total alleles for brown fur, the 120 that we calculated up here, and we can divide by the grand total, 200, and we get 0.6. Now notice, if we add 0.4 and 0.6, what number are we going to get? 0.4 plus 0.6? Correct. We're going to get 1. Well, remember what we said. 1 is 100%. So if we only have 2 alleles, they should add up to 1 when we double check our work. Now, keep in mind, if we have weird numbers, and in some of the problems we work, we will. You've all worked math problems. You might get that one of these is 0.41 and one of these is 0.60, depending on how we do our rounding. So you might get 0.99 or 1.01, but it should be very close to 1. Like I say, it might be off a little depending on rounding. Now, there's another way that we could do this. As soon as we calculate one of the alleles, so suppose as we're working a problem like this, we go through and we figure out that black fur is 0.4. That's the frequency. The other way we can do this is, since we know black fur, we can take 1 minus the frequency for black fur, and that tells us the frequency for the other allele, because we know the 2 of them have to add up to 1. So if you think about this mathematically, if we know that, the allele for black fur and the allele for brown fur equal 1, and we want to find black fur, and we already know what brown fur is, we can subtract the brown fur from both sides. So once we know one of the alleles, we can always figure out the other allele by subtraction. Now, in this case, we can do this as soon as we find one allele. But let's go back to this first gene pool that we showed. Notice here we have 3 alleles. We have X1, X2, and X3. Here we can figure out what the last allele is, but before we can figure out the last allele, say if we're going in numerical order, before we can figure out X3, we have to figure out X1 and X2. When we get to the last allele, whether it's the second allele or the third allele, or maybe there's 10 alleles in the population, when we get to the 10th allele, we could always figure it out by subtraction, 1 minus the frequency of all the other alleles. The problems that we work, as I say, mathematically, things get much more complicated if we talk about more than one gene, or if we talk about more than 2 alleles. So we'll be working problems with 2 alleles. So when we figure out one allele, we can always figure out the other allele by subtraction. Now, going back to what I said earlier, the first allele that we're going to figure out is we're going to figure out the recessive allele. We're going to look at individuals with the recessive phenotype. We're going to figure out the recessive allele. And then the way we're going to figure out the dominant allele is 1 minus the recessive allele. And that's because of what we said earlier. If we think about this population of mice, I can look at a mouse and I can see that it has brown fur. And notice that if a mouse has brown fur, there's only one genotype that's possible for this mouse. On the other hand, when I look at this population of mice and I see the ones with black fur, some of them are heterozygous, some of them are homozygous. I don't know how many are heterozygous and how many are homozygous. So mathematically, it's very difficult to figure out the allele for black fur and be correct. So let's look at an alternative way to do this. And keep in mind that a lot of this we're going to be looking at in more detail. So this is really just an introduction, so don't panic. And hopefully you feel that math is your friend, because we're definitely going to have to use a lot of math to solve Hardy-Weinberg problems. So let's think about these mice that have a brown fur phenotype. We've said that their frequency is 0.36, because remember what I said earlier, frequency is really a percentage. So 36% is 0.36, 92% would be 0.92. Now we want to keep this number in mind. So we'll come back to this in a few minutes. So I'm going to copy it because we're going to use it a little bit further down. So let's see what the Hardy-Weinberg equation is now, because we're going to be using these frequencies in the Hardy-Weinberg equation. The Hardy-Weinberg equation was developed in 1908. So remember, Mendel's work was rediscovered in 1900. So this is really just a few years, within a decade after Mendel's work was discovered. And it was discovered separately by two scientists, Hardy, who was an Englishman, and Weinberg, who was a German. And what they looked at was the fact that let's take two variables, think of this like an algebra equation. Let's take p plus q and let's square that. And if we do, our result is p squared plus 2pq plus q squared. And what Hardy and Weinberg both said, they were working on this separately. Let's think about p being the dominant allele and q being the recessive allele. I have no idea why they chose p and q. Actually, that was chosen by geneticists before them, but I have no idea why the original geneticists decided to use p for the dominant allele, q for the recessive allele, but that's fairly standard notation in a lot of genetics. Well, what Hardy-Weinberg said was, if you think about taking p plus q and squaring it and looking at the results, that's really the same thing that we see if we look at a Punnett square, where we cross two heterozygotes. So let's think about having a female whose genotype is PQ, she's heterozygous. So remember that that female will be able to make two types of eggs. Half of her eggs will be P, half of her eggs will be Q. And let's cross this female with a male whose genotype is also PQ. So half of his sperm will be P, half of his sperm will be Q. So remember how we fill out a Punnett square. We carry the alleles from the female down to each cell underneath. We carry the alleles from the male across to each cell. And if you remember, whenever we cross two heterozygotes, we get a similar pattern. the pattern that we see for our genotypes. We're not going to talk about phenotypes for now. We'll get to that eventually. But the pattern that we see for our genotypes is that a fourth of the offspring will be PP. 2/4 of the offspring will be PQ. And 1/4, that last cell in the lower right, will be QQ. Well, what Hardy-Weinberg said was, okay, notice PP is really, if you think about multiplying those, is really P squared. We've got 2 individuals that are PQ. Well, that's the middle term in the expansion of this algebraic expression above. And QQ, if we think about multiplying them, is really Q squared. So what Hardy and Weinberg were able to show, and they went through some very rigorous mathematical proofs, is that you can use an expression like this to study things about a population. And what we're going to think about in the problems we work is that P is going to be the frequency of the dominant allele, and Q is going to be the frequency of the recessive allele. Although in the problems we work, if you'd rather use, big A and little a or something like that, whatever is appropriate for the problem, you can certainly do that. Just like in 105, it doesn't really matter what letters we use to designate the dominant allele and recessive allele. But keep in mind, if you would go online to any place that's telling you about Hardy-Weinberg, you're probably going to see P's and Q's. Now, when we look at these results, this P squared plus 2PQ plus Q squared, we can use these results to find out if our population is in something that we call Hardy-Weinberg equilibrium. And in a population that's in Hardy-Weinberg equilibrium, probably the best way to think about it is that the allele frequencies are not changing. So with our mice up here, when we did a little bit of math, we concluded that the allele for black fur has a frequency of 0.04. The allele for brown fur has a frequency of 0.06. If this population is in Hardy-Weinberg equilibrium, we should be able to come back in a few generations and the frequency of the allele for black fur will still be 0.4. The frequency of the allele for brown fur will still be 0.6. And not only that, we can use these frequencies to give us some other information. So if you think about P squared is individuals who have 2 copies of the dominant allele. So in other words, P squared is our individuals that are homozygous dominant. Those 2 cells that are PQ, those 2 cells represent individuals that are heterozygous. And then finally, this last cell down in the lower right-hand corner, Q squared. Q squared is all the individuals in this population that are homozygous recessive. Now, think about some of the things that we know from genetics. So remember, one of the things we talked about towards the beginning of this discussion, and that I'm sure you talked about back in 105. If we combine the individuals who show the dominant allele, who are homozygous dominant, and the individuals who show the dominant allele because they're heterozygous, then if we add these 2 together, this is everybody who has a dominant phenotype. So we can figure out the dominant phenotype by adding P squared and 2PQ. Similarly, Q squared tells us all the individuals that have the recessive phenotype. And that's really why we're always going to start with the recessive phenotype, because remember what I said above. In this case, for these mice, if you have brown fur, you have to be homozygous recessive. And notice we can figure out lots of other things. Suppose I asked you what frequency of the mice in this population have at least one recessive allele. So think about who has one recessive allele. So notice individuals who are p-squared, they don't have a recessive allele, but individuals who are 2pq, they have a recessive allele. Individuals who are q-squared, they have a recessive allele. So if we add up these two categories, this tells us how many individuals in this population have a recessive allele. And finally, we'll spend more time with this next week, but let's think about why we have to start with individuals who have the recessive phenotype. So let's go back to our population here. So let's think about this is the frequency of individuals with brown fur, 36%, which we said expressed as a frequency is 0.36. Now we know that brown fur, going back down here, we know that these individuals are q-squared. We know that this is their phenotype. So suppose I gave you this chart, but this chart, I didn't have the homozygous black mice separated from the heterozygous black mice. How could you find out how many individuals have black fur? Well, step out of genetics for a moment and think about this as an algebraic equation. Suppose you had an equation and the equation was just q-squared equals 0.36 in a math class. And the question was, solve for q. What would you have to do to find out what q equals mathematically?
Square root.
Would you take the square root of 0.36?
Take the square root of both sides of the equation. So take the square root of q squared, that's just q, and then take the square root of 0.36, which is, guess what? 0.6, the same number that we came up with here. Now let's think about why we can't start out with individuals that show the dominant characteristic. So remember, usually when we look at a population, we won't know the genotype. They gave it to us in this example, but usually all we'll know is the phenotype. So let's look at all the mice who have black fur. So 48% plus 16%, 64%. So black fur is equal to 64%, or if we put that as a frequency, it's 0.64. So suppose you try to do the same thing and find the dominant allele. So now we have, okay, it's not good. I'm seeing a spinning beach ball, so I don't know if Microsoft Word is going to crash on me. So suppose you try to solve this. And what you do is okay. I'm going to kill Microsoft Word and just open a new document. So let's share our screen again, but let's go to the whiteboard. Okay. So suppose we decide to solve this by solving for the dominant allele. And we say that if we look at the dominant allele, p squared equals 0.64. Well, this is the same equation mathematically. The numbers are different, the variables different, but we're going to solve it the same way. So if we take the square root of both sides, if we take the square root of p squared and the square root of 0.64, what do we get? P equals 0.8. Well, if we go back and we look at our document again, notice that when we figured this out, P, the allele frequency for black fur, was not 0.8, it was 0.4. So we don't get the right answer. And the reason we don't get the right answer is if we take the square root, that's assuming that every individual in that population that has black fur is homozygous dominant. And we know that they're not all homozygous dominant. So from that point of view, this is not going to give us an accurate answer. So we want to make sure that we always start with the recessive phenotype and use that to find the recessive allele. Now, depending on what information we're given, we may have to do a step or two to figure out how many individuals, what frequency show the recessive phenotype. But once we figure that out, that's where we want to use that information to find the recessive allele frequency. So are there any questions so far about this? And keep in mind, next week, we're going to talk about this some more, and we're going to work lots of examples, probably so many that you'll be tired of them. But so later today, I will post another short assignment, and this one will be asking you some questions about genetics vocabulary. So some of the basic terms that we talked about in Biology 105, genotype and phenotype, dominant and recessive, genotype, phenotype, dominant, recessive, what did I miss? Heterozygous and homozygous. And I'll have you work a monohybrid cross. So that will be due on Wednesday, right before we come to class. So most of our assignments are due, at midnight someday, or right before midnight, I guess I should say. This one will be due at 8:59 in the morning on Wednesday. So I'll have that up later. Are there any questions before we leave? And I let you have some peace for a whole weekend and a whole weekend plus one day, actually. Okay, if there's no questions, you can go ahead and go enjoy the extra day off. And I will see you on Wednesday morning.
Okay, take care.
Thank you. Have a good
too.
day.
Bye-bye, you too.
Yeah.
Bye.
Stay safe.
You too.